Have you ever just, stopped to think about how incredible the human body is? Like, really appreciate it as this amazing self-regulating machine.
It really is packed with wonders, many working away without us even noticing.
Exactly. And today, we're diving deep into one of those vital, maybe sometimes unsung systems, the urinary system.
Ah, yes. Much more than just plumbing.
Definitely. We're going to pull back the curtain, explore its fascinating microscopic world, and we'll be drawing our key insights from the fantastic resource, Histology, a text and atlas 8th edition.
A classic text. Great visuals in there, too.
Right. So our mission today is pretty clear. We want to quickly get to the heart of it, find those really important nuggets of knowledge.
Give you those real aha moments.
Precisely. We'll look at the intricate microscopic anatomy, the cells involved, the different tissue types.
And really try to paint a picture of what this looks like under a microscope.
Yeah, connecting structure to function, maybe touching on some relevant molecular bits too. It's all about getting you the core insights quickly and thoroughly.
All right, let's get started. Where should we begin?
Okay, let's unpack this system, starting with the main players.
Sounds good. So, fundamentally, you've got the paired kidneys. They're the star players doing the filtering.
Right, one on each side usually.
Then the ureters, also paired. These tubes carry the urine down from the kidneys.
To the bladder.
Exactly. The urinary bladder, which is essentially a storage tank. And finally, the urethra, the tube that takes urine from the bladder out of the body.
Simple enough lineup?
Mm-hmm.
But the kidneys themselves,
Yeah.
they do more than just filter waste, right?
Oh, much more. People often underestimate them. They're crucial for maintaining your body's overall balance homeostasis.
How so?
Well, they conserve vital fluids and electrolytes, they help regulate your plasma pH, keep your blood volume stable, and, surprisingly to many, they function as endocrine organs, too.
Endocrine, like hormones.
Yep, they make erythropoietin, or EPO. You've probably heard of that.
Stimulates red blood cell production.
That's the one. Signals bone marrow when oxygen is low.
Yeah.
They also produce renin, which is absolutely key for blood pressure control.
Okay.
And they perform a critical step in activating vitamin D, converting it to calcitriol, which you need for calcium metabolism. So yeah, a real multitasker.
That's quite the resume for an organ we mostly just associate with bathroom breaks.
It really is.
So, structurally, when we zoom in on the kidney itself, what does this powerhouse actually look like?
Okay, let's picture it. The kidney's wrapped in a capsule, a connective tissue covering. It actually has two layers.
Two layers.
Yeah, an outer fibrous layer and an inner one with myofibroblasts. These inner cells can contract, though we're still figuring out exactly why that's important.
Interesting. And inside the capsule?
You see two main regions if you cut it open. The outer part is the cortex. It's reddish-brown and gets almost all the blood flow, like 90–95%.
Wow, okay.
This cortex is where you find the renal corpuscles, those little filtration units we'll talk more about, and lots of winding tubules. Then deeper inside, you have the medulla. It's lighter in color.
And how do they relate?
Well, you see these sort of vertical stripes projecting from the medulla up into the cortex. Those are called medullary rays.
Okay.
They're basically bundles of straight tubules and collecting ducts expanding outwards. The cortical tissue between these rays is called the cortical labyrinth.
Labyrinth, I like that. And the medulla itself?
The medulla is mainly made of these straight tubules, collecting ducts, and a special capillary network called the vasa recta.
Vasa recta, got it.
These structures in the medulla are organized into cone-shaped structures, the renal pyramids. Humans usually have around 8 to 18 of them.
Pyramids.
Their wide bases face the cortex, and the pointy tips, called papillae, stick into these little cup-like structures called minor calyces.
Calyces, like cups.
Exactly. And the very tip of the papilla, called the area cribrosa, is like a sieve. It's got tiny holes where the collecting ducts empty the urine into the calyces.
Fascinating. You also mentioned renal columns.
Ah, yes. Renal columns, or columns of Bertin. These are actually bits of cortical tissue that extend down between the pyramids into the medulla. So, anatomically, they're considered part of the medulla, even though they contain cortical structures.
A bit confusing, but okay.
Yeah, it maps out functionally. Think of it this way. Each pyramid, along with the cortical tissue sitting over its base and along its sides, including those columns, forms a kidney lobe.
Well,
And then an even smaller unit, the lobule, is basically one of those medullary rays plus the cortical labyrinth tissue surrounding it. That's kind of the basic secretory unit.
it's incredibly organized. And you said almost all the blood goes to the cortex. How does that complex blood supply work?
It's a really intricate vascular system, absolutely critical for its function. Blood comes in via the renal artery.
Right.
That branches into interlobar arteries, which run up between the pyramids. Then they curve over the bases of the pyramids as arcuate arteries.
Arcuate, like an arc.
Exactly. And off these arcuate arteries come the interlobular arteries radiating outwards into the cortex.
Okay, getting smaller and smaller.
Precisely. And from these interlobular arteries, you get the afferent arterioles. Each one leads into a glomerulus.
The filtration unit.
Yes, that capillary tuft where filtration starts. Now, here's a key difference from most places in the body. Blood leaves the glomerulus not through a venule, but through an efferent arteriole.
Arterial in, arterial out.
You got it. And what that efferent arteriole does next depends on where the glomerulus is.
Oh.
If it's a cortical glomerulus, the efferent arteriole breaks up into a dense network of capillaries surrounding the tubules in the peritubular capillaries.
Okay.
But if it's from a glomerulus near the medulla, a juxtamedullary glomerulus, the efferent arteriole forms the descending vasa recta. These are long, straight vessels that go deep into the medulla, running alongside the loops of Henle.
Crucial for that concentration mechanism later, I bet.
Absolutely essential. Then, the venous blood collects and generally follows the arterial path back out. There's also lymph drainage and nerve supply, mostly sympathetic nerves running with the vessels, especially the afferent arterioles. They help regulate filtration rate.
But you mentioned transplants.
Right. Interestingly, a transplanted kidney functions fine without those external nerves, showing the kidney has a lot of intrinsic control.
Amazing. Okay, let's pause there for a moment. The basic structure and blood flow are set.
A good foundation. And thank you for being part of the Last Minute Lecture family.
All right, now for the really cool part, I think. The nephron. You called it the fundamental unit.
That's right. The nephron is the structural and functional powerhouse of kidney. Remember those numbers?
Yeah.
About 2 million per kidney.
Incredible.
These are what actually produce the urine. The collecting ducts, which come slightly later in the path, handle the final concentration steps.
So what makes up a nephron?
It's essentially got two main parts. First, the renal corpuscle. Think of it as the starting point, the filter. It's this spherical structure containing the glomerulus, that tuft of capillaries, maybe 10 to 20 loops, all wrapped up in the Bowman's capsule, which is a double-layered epithelial cup.
Double-layered.
Yep. And the corpuscle has two poles. The vascular pole is where the efferent arteriole enters and the efferent arteriole exits.
Makes sense.
The other end is the urinary pole. That's where the filtered fluid, the ultrafiltrate, leaves the capsule and enters the first part of the nephron's tubule system, the proximal convoluted tubule.
Got it. So corpuscle, then tubules. What are the tubule parts?
It's a long, winding road. You have the proximal thick segment, which starts convoluted, PCT, and straightens out. Then comes the thin segment. That's the famous loop of Henle.
The loop.
After the loop, you get the distal thick segment, again, starting straight and then becoming convoluted, DCT. So proximal tubule, loop of Henle, distal tubule. That's the core nephron.
And you mentioned different types of nephrons.
Yes, based mainly on location and loop length. Subcapsular or cortical nephrons are mostly in the outer cortex and have short loops that barely dip into the medulla.
Okay.
Then you have juxtamedullary nephrons. Their corpuscles are near the medulla border, and they have these really long loops of Henle that go deep, deep into the medullary pyramids.
And those are important for
concentrating the urine. Absolutely critical. There are also intermediate or mid-cortical ones in between.
Okay, so we have the structure. How does that renal corpuscle, that tiny filter, actually clean the blood? It seems like such a delicate process.
Delicate yet powerful. It all happens across the glomerular filtration barrier within the renal corpuscle. It's made of 3 key layers.
3 layers.
First, the endothelium of the glomerular capillaries. These cells are full of pores or fenestrations, but unlike pores and many other capillaries, these ones don't have diaphragms closing them off.
So very leaky.
Very permeable to water and small solutes, yes. Plus, they have lots of aquaporin 1 water channels, AQP1, making water movement super efficient.
Layer 1, leaky endothelium. What's next?
Layer 2 is the glomerular basement membrane, the GBM. This is a thick, specialized basal lamina sandwiched between the endothelium and the next layer of cells.
Thick, you say?
Yeah, unusually thick, around 370 nanometers. It's jointly made by the endothelial cells, and the podocytes, those are the next layer. The GBM is rich in specific types of type IV collagen, laminin, proteoglycans. It's a complex matrix.
And important, I gather.
Critically, Problems here cause major issues. Like in Alport syndrome, mutations in a specific type IV collagen gene lead to a faulty, often thickened GBM, causing blood in the urine, protein loss, and eventual kidney failure.
Wow.
Or Goodpasture syndrome, that's an autoimmune disease where the body attacks the GBM, leading to rapid kidney damage and often lung bleeding too. Very serious.
Okay, so endothelium GBM, what's layer 3?
Layer 3 is the visceral layer of Bowman's capsule, formed by highly specialized cells called podocytes.
Podocytes, foot cells.
Exactly. They have these major processes, and then extending from those are secondary processes called pedicels, or foot processes. They look like little feet resting on the GBM.
Okay.
And the cool part is the foot processes of one podocyte interlock, like fingers, with the foot processes of its neighbors.
Leaving gaps.
Precisely. These elongated gaps between the interdigitating pedicels are called filtration slits. And spanning across each slit is an ultra-thin diaphragm, the slit diaphragm.
Like a final filter.
Yes, the ultimate size-selective barrier. It's a complex protein structure, kind of like a zipper. A key protein here is nephrin.
Nephrin.
Mutations in the gene for nephrin cause a severe condition called congenital nephrotic syndrome, where this filter breaks down, leading to massive protein loss right from birth.
So it's endothelium, GBM, and the podocyte slit diaphragm working together.
Exactly. Plus, there's a negatively charged glycocalyx on the endothelial surface and a space under the podocytes that also play roles. The GBM acts as a physical barrier and an charge barrier, repelling negative proteins like albumin, while the slit diaphragm is the fine sieve based on size.
How does it not get clogged?
Good question. The negative charges help repel things, and then there are special cells called mesangial cells tucked within the glomerulus that act like little vacuum cleaners.
Mesangial cells. Tell me about those.
Right. They sit in the spaces between the glomerular capillaries sharing the GBM. They're embedded in their own matrix material.
What do they do?
Several things. They provide structural support. They're phagocytic, so they engulf trapped residues and immune complexes, keeping the filter clean.
Like the vacuum cleaner you mentioned.
Exactly. They also secrete various signaling molecules, and they have contractile properties, which might help regulate blood flow or respond to stretching.
And are they involved in disease too?
Oh, definitely. Overproliferation of mesangial cells is a hallmark of several important kidney diseases, like IgA nephropathy, lupus nephritis, and diabetic nephropathy. Their response is often part of the disease process.
Incredible complexity in such a tiny space. So how does this whole unit, this glomerulus, communicate with the rest of the body about things like blood pressure? You mentioned renin earlier.
Ah, yes. That brings us to the juxtaglomerular apparatus, or JGA, a very clever sensor system right at the vascular pole of the renal corpuscle.
JDA. Okay, what's it made of?
3 key components. First, the macula densa. This is a patch of specialized cells in the wall of the discal tubule, right where it loops back up near its own glomerulus.
Macula densa. Dense spot.
Kind of. The cells look different, narrower, taller, with crowded nuclei. Their job is to monitor the sodium concentration in the fluid passing through the tubule at that point.
Sensing the salt level. Okay, component 2.
Juxtaglomerular cells, or JG cells. These are actually modified smooth muscle cells in the wall of the afferent arteriole, the vessel bringing blood into the glomerulus.
Modified how?
They contain granules packed with that enzyme, renin.
Ah, the source of renin.
Exactly. And the third component is a group of cells called extraglomerular mesangial cells, or LACES cells, which sit in the angle between the afferent and efferent arterioles and the macula densa. Their role is less clear, but they're part of the structure.
So macula densa senses salt. JG cells have renin. How does it work for blood pressure?
It's the heart of the renin-angiotensin-aldosterone system, or RAS. If the macula densa senses low sodium, which often implies low blood pressure or volume, or if the JG cells directly sense low pressure in the afferent arteriole,
They release renin.
bingo. Renin gets into the blood and starts a cascade. It cuts a protein called angiotensinogen, made by the liver, into angiotensin I.
Okay.
Angiotensin then travels mainly to the lungs, where another enzyme, angiotensin-converting enzyme, ACE, converts it to angiotensin II.
Angiotensin II, that's the active one.
That's the big player. It's a powerful vasoconstrictor. It squeezes blood vessels, raising pressure immediately. And it also stimulates the adrenal glands to release aldosterone.
Aldosterone. What does that do?
Aldosterone acts back on the kidney tubules, mainly the distal parts and collecting ducts, telling them to reabsorb more sodium and water and secrete potassium. More sodium and water reabsorption means increased blood volume, which also raises blood pressure.
A whole feedback loop. Macula densa senses, JG cells release renin, leads to angiotensin II and aldosterone, which fix the low-pressure sodium problem.
You've got it. The macula densa provides feedback to the JG cells and can also influence the local glomerular filtration rate directly. It's incredibly elegant.
And ACE inhibitors, they block that conversion step.
Exactly. They block the ACE enzyme, so less angiotensin II gets made, less vasoconstriction, less aldosterone, lower blood pressure. They were a huge breakthrough, originally derived from snake venom, actually.
Fascinating stuff. The kidney isn't just filtering, it's actively sensing and regulating the whole body.
Precisely. It's a dynamic, responsive organ. And again, thank you for being part of the Last Minute Lecture family.
Now that we understand filtration and how the kidney senses the body's needs, let's follow that filtrate. What happens as it goes through those long, winding tubules? You said it gets modified.
Extensively modified. That initial glomerular ultrafiltrate is basically plasma minus the big proteins and cells. As it flows through the nephron tubules and then the collecting ducts, the kidney selectively reabsorbs what the body needs: water, sodium, glucose, amino acids, both actively and passively.
Pulling the good stuff back.
Right. And it also actively secretes certain waste products into the tubule, things like creatinine, certain drugs, organic acids, and bases.
Fine-tuning the composition.
Exactly. The end result is that the initial large volume of filtrate gets drastically reduced, and the final urine becomes much more concentrated or hyperosmotic compared to blood.
This concentration relies heavily on that countercurrent multiplier mechanism we hinted at earlier.
Okay, let's trace the path. First stop after Bowman's capsule. The proximal convoluted tubule, the PCT, this is the workhorse of reabsorption. About 65% of everything filtered water, sodium, potassium, etc. gets reclaimed right here.
65%. How do the cells manage that?
They're built for it. They're cuboidal cells with a very prominent brush, border-dense, long microvilli on the luminal side that massively increase the surface area.
The rush border. Okay.
They also have lots of folds on their basal side and are packed with mitochondria, especially near the base, providing the ATP energy needed for all the active transport.
Like the Na plus K plus pump.
And absolutely, that pump is a key driver. Water follows passively, largely through those AQP1 channels. Glucose is completely reabsorbed here, using sodium-glucose cotransporters, SGLT2 mainly. Amino acids, too, almost all recovered. Even small proteins get pulled in and broken down.
So reclaiming almost everything valuable.
Pretty much. The PCT also plays a role in acid-base balance by reabsorbing bicarbonate and secreting some acids.
Then it straightens out.
Yes, into the proximal straight tubule, or thick descending limb. Still some reabsorption here, including any remaining glucose via a high-affinity transporter, SGLT1. The cells are a bit shorter, brush border, less developed.
And then the loop of Henle's thin segment.
Right. This is where things get really interesting for concentration. There are actually different types of epithelial cells along the thin segment, varying a bit in structure.
But the key is permeability, right?
Exactly. The thin descending limb is very permeable to water, lots of aquaporins, but not very permeable to salts. So as it dips into the salty medulla, water leaves passively, concentrating the fluid inside the tubule.
Makes sense. Water out, stuff inside gets concentrated.
Then the thin ascending limb flips the script. It's highly permeable to sodium and chloride, which move out into the interstitium, some active, some passive transport, but it's essentially impermeable to water.
So salts leave, but water stays in.
Correct. This dilutes the tubular fluid, making it less concentrated than the surrounding interstitium. That's why it's called the diluting segment. This movement of salts out, without water following, is crucial for building up the saltiness of the medulla.
Okay, that's the countercurrent multiplier starting to take shape. What about that protein you mentioned, uromodulin?
TEM horsefall protein, or uromodulin. It's actually made primarily in the next segment, the thick ascending limb. It gets secreted into the tubule lumen.
What does it do?
It seems to help with NaCl reabsorption and urinary concentration, acts as a defense against UTIs by trapping bacteria, and might help prevent kidney stone formation.
Yeah.
But in certain diseases, it can form the matrix of urinary casts, which we see in urinalysis.
Got it. So after the thin ascending limb
comes the distal straight tubule, also called the thick ascending limb, TAL. This continues pumping ions, sodium, potassium, chloride out into the interstitium, again without water following, still diluting the tubular fluid.
More salt removal.
Yes, and this ion movement also helps drive the reabsorption of calcium and magnesium in this segment. The cells here are large, cuboidal, packed with mitochondria for power.
Then the distal convoluted tubule, the DCT.
Right. Shorter, less tortuous than the PCT, no real brush border. This is where fine-tuning happens. It's the main site for calcium reabsorption, regulated by parathyroid hormone.
Hormonal control.
Yes. Also, more sodium and chloride reabsorption. This is where thiazide diuretics work. Potassium secretion, hydrogen secretion to acidify the urine, and ammonium secretion. It has the highest Na plus K plus AT pace activity anywhere in the nephron.
A lot of final adjustments.
Indeed. Then there's a short connecting tubule, CNT, which links the nephron to the collecting duct system. It's important for potassium secretion, influenced by aldosterone.
And finally, the collecting ducts.
Yes, the cortical and medullary collecting ducts. These run down through the cortex and medulla, gathering fluid from multiple nephrons. This is where the final urine concentration is determined, under the control of ADH.
ADH again.
Right. The collecting ducts have distinct cell types. The main ones are principal cells. They're pale staining and have those crucial AQP2 water channels on their apical surface. ADH controls whether these channels are inserted into the membrane.
So ADH presents water channels open.
Essentially, yes. ADH causes AQP2 to move to the membrane, making the duct permeable to water. Water then leaves, following the osmotic gradient into the hypertonic medulla, concentrating the urine. Principal cells also have receptors for aldosterone, making them key sites for sodium reabsorption and potassium secretion regulated by that hormone.
And the other cell type?
Dark cells, or intercalated cells. Fewer of these, denser cytoplasm, lots of mitochondria. They handle acid-base balance. Alpha intercalated cells secrete acid, H+, while beta intercalated cells secrete bicarbonate base.
So the collecting duct isn't just a passive pipe. It's actively managing water and acid base.
Absolutely. And it's crucial to understand that aldosterone's main action for sodium-potassium balance is really on these principal cells in the CNT and collecting ducts, not so much the DCT as previously thought.
Okay, let's tie this together. The countercurrent systems.
Right. You have two main parts. The countercurrent multiplier is the loop of Henle. The descending limb loses water, concentrating the fluid. The ascending limb pumps out salt but keeps water, diluting the fluid and making the medulla salty. This creates the gradient.
Multiplies the saltiness down deep.
Exactly. Then you have the countercurrent exchanger, that's the vasa recta, those long capillary loops running alongside the loops of Henle.
What do they exchange?
As blood flows down into the salty medulla, it picks up salt and loses water. As it flows back up towards the cortex, it loses salt and picks up water. The hairpin turn and slow flow allow this exchange without washing out the medullary osmotic gradient created by the loops. It preserves the gradient while supplying blood. And finally, the collecting duct is the osmotic equilibrating device. As it passes down through that salty gradient established by the loop and maintained by the vasa recta, ADH determines its water permeability. High ADH means high permeability, water leaves, urine gets concentrated, equilibrating with the medulla. Low ADH, low permeability, water stays in, urine stays dilute.
So aquaporins and hormones are the keys to that final step.
Absolutely central. Those AQP2 channels controlled by ADH are the gatekeepers for water reabsorption in the collecting ducts.
You mentioned AQP1 earlier in the PCT.
Yeah.
Any others?
Yes, AQP3 and AQP4 are found on the basolateral side of the collecting duct cells, providing the exit pathway for water into the interstitium, a whole family of water channels.
Yeah. And ADH regulation. It responds to dehydration.
Primarily, yes. If your plasma osmolality increases, you're dehydrated, or if your blood volume pressure drops significantly, your brain releases ADH.
Which tells the kidneys to save water.
Exactly. By inserting AQP2 into the collecting duct membranes, if you drink lots of water, ADH levels drop, AQP2 is removed from the membrane, the ducts become impermeable to water, and you excrete dilute urine.
And problems with ADH or AQP2 cause?
Diabetes insipidus. Either the brain doesn't make ADH, central DI, or the kidneys can't respond to it, nephrogenic DI, sometimes due to AQP2 mutations. Either way, you can't concentrate urine and produce huge volumes of dilute urine.
Okay. And what about checking the final product, urinalysis?
A cornerstone of kidney diagnostics. Simple, non-invasive. We measure pH, specific gravity, which reflects concentration, look for abnormal things like glucose, diabetes mellitus, bilirubin, liver issues, ketones, diabetes, starvation, blood, and especially protein.
Proteinuria is a big red flag.
Significant proteinuria, especially albuminuria, often points to glomerular damage, that filtration barrier breaking down. Those small amounts can appear temporarily after intense exercise or dehydration.
And those casts you mentioned.
Urinary casts, yeah. They're cylindrical molds formed in the tubules, usually made of that Tam-Horsfall protein matrix. If they contain red blood cells, white blood cells, or epithelial cells, it tells you there's inflammation or damage happening within the kidney itself. Very informative.
A simple test giving complex information.
It really is a window into kidney function. And with that, thank you for being part of the Last Minute Lecture family.
So we followed the journey. Filtration, massive reabsorption and secretion in the tubules, concentration via countercurrent mechanisms and hormones. Now, this concentrated urine needs to get out. What about the exit route?
Right. After leaving the collecting ducts at the papilla tip, the area cribrosa, urine flows into the minor calyces, then major calyces, the renal pelvis, which is like a funnel within the kidney, and then down the ureters to the bladder for storage, and finally out via the urethra.
And these passages just transport it, no more modification.
Correct. Their job is transport and storage, not changing the urine composition. Structurally, most of these passages, calyces, pelvis, ureters, bladder, share a common plan. A mucosa lining the inside, then a layer of smooth muscle called the muscularis, and an outer connective tissue layer, the adventitia, or sometimes serosa on the bladder. Importantly, they lack a muscularis mucosa or a distinct submucosa, unlike the GI tract.
And the lining, the mucosa, is special?
Very special. It's lined by transitional epithelium, also called urothelium.
Urothelium. Why is it special?
Its main superpower is being incredibly impermeable to water and salts. You don't want urine leaking back into the body tissues.
Makes sense. What does it look like?
It's stratified, meaning multiple layers. The key feature is the surface layer of large, often dome-shaped cells called umbrella cells. They bulge into the lumen when the bladder or ureter is relaxed.
Umbrella cells.
Yeah. And when the bladder fills and stretches, these umbrella cells can dramatically flatten out and spread, allowing the epithelium to accommodate the increased volume without breaking its barrier function.
Oh, wow. Stretchy and impermeable.
Exactly. The cell membranes themselves have unique features contributing to this. The apical surface of the umbrella cells is covered in rigid-looking patches called urothelial plaques.
Plaques?
Yes, made of proteins called uroplakins arranged in a crystalline structure. This forms an asymmetric unit membrane, AUM, that's thought to be key to the impermeability.
Okay.
And when the bladder needs to expand, the cell inserts more of this AUM membrane from internal storage vesicles, fusiform vesicles, into the surface. When it empties, the excess membrane is pulled back in. It's incredibly dynamic.
So it's not just a passive barrier.
Not at all. We now know the urothelium is active in signaling. It can sense stretch, chemicals in the urine, and communicate with nerves and muscles in the bladder wall. It even produces neurotransmitters.
That's amazing. A barrier that talks back.
Sort of, yeah. And clinically, this is relevant for UTIs.
Yeah.
Some bacteria, like uropathogenic E. coli, have adhesins that specifically bind to these uroplakins, helping them stick to and invade the bladder lining.
Yeah, chink in the armor.
Potentially, yes.
What about the muscle layer that pushes the urine along?
The muscularis is smooth muscle, arranged to produce peristaltic waves in the ureters to propel urine down. Interestingly, in the ureters, the typical arrangement is inner longitudinal muscle and outer circular muscle, kind of the opposite of the gut wall.
Inner longitudinal, outer circular. Okay.
Let's quickly trace the passages. The ureters run from the kidney pelvis down to the bladder, about 24 centimeters long.
Right.
They enter the bladder wall at an oblique angle. This is important because when the bladder fills and pressure rises, it compresses the ends of the ureters, acting like a valve to prevent urine refluxing back up to the kidneys.
Smart design.
Very. The ureter wall has that inner longitudinal and middle circular smooth muscle, and sometimes a third outer longitudinal layer appears near the bladder.
And the bladder itself.
A highly distensible muscular sac. The main muscle is the detrusor muscle. When it contracts, you urinate.
Detrusor. Okay.
At the junction of the bladder and urethra, the smooth muscle forms an involuntary internal urethral sphincter.
Involuntary.
Yes. Micturition, or urination, involves complex neural control. Parasympathetic nerves cause the detrusor to contract and the internal sphincter to relax. Sympathetic nerves generally do the opposite. Relax the detrusor, tighten the sphincter, promoting storage.
And we have voluntary control, too.
Yes, via the external urethral sphincter, which is made of skeletal muscle and controlled by the pudendal nerve. That's the one you consciously control.
Got it. And finally, the urethra.
The exit tube. Different in males and females. In males, it's longer, about 20 centimeters, and passes through the prostate gland. prostatic urethra lined by urothelium, then a short membranous part surrounded by the external sphincter, changing epithelium, and finally, through the penis, spondy urethra, mostly pseudostratified columnar, becoming stratified squamous at the tip.
Okay.
In females, it's much shorter, 3 to 5 centimeters. Starts with urothelium near the bladder, then changes to stratified squamous or pseudostratified columnar. The shorter length is one reason UTIs are more common in women. Both sexes have small glands associated with the urethra.
A comprehensive tour of the exit path.
It completes the journey from filtration to elimination. And thank you, once again, for being part of the Last Minute Lecture family.
Wow, what an incredible system. The complexity, the interconnectedness, it's just astounding.
It truly is. From the molecular details of nephrin holding the filtration slits together, to the physics of the countercurrent multiplier building that osmotic gradient, all the way to the urothelium dynamically stretching in the bladder, every level matters.
And understanding those microscopic details really illuminates the bigger picture of how the body maintains balance and what happens in disease.
Absolutely. Structure and function are inseparable here.
It really has been quite a journey following that fluid from plasma filtrate to highly regulated concentrated urine.
Okay.
It just highlights the body's remarkable ability to constantly adjust and maintain that internal equilibrium.
Homeostasis in action.
It really makes you think. What other seemingly simple body processes, things we take for granted, hide that same kind of profound microscopic detail and intricate regulation?
That's a great question to ponder. There's always more to explore beneath the surface.
Definitely. Keep that curiosity alive, everyone, and keep doing your own deep dives. Thank you so much for joining us today as we explored the urinary system.
Yes, thank you for tuning in. It's been a pleasure.